Welcome back as we learn about the cell. In this section, we're going to learn all about the plasma membrane. So first, I want to refresh your memory that structure reflects function as we're learning about the cell. So as we have a different structures, and we're going to have different functions. And so my learning tip for learning about the cell is you do need to learn the functions for all parts of the cell as listed on not only this section, but the next several sections on the cell. And please learn the functions that I am providing for you. If I state more than one function for a structure, you need to learn more than one because these are the functions that we're going to be talking about the entire semester. So yes, when we get to the organ systems, we're still going to be talking about, oh, what's the organelle that's going to be doing this function for this cell? So if I say, like this, this, the liver detoxifies, then you would know, oh, then the liver has a lot of, and you would know what organelle, but the liver cells would have a lot of. Okay, so let's talk about our friend, the eukaryote. So here is just a drawing of a random eukaryotic cell. Not a great drawing. Like I wouldn't copy it and expect to get credit for it because it's not perfect. I'm just throwing it out there. It's just to fill up space on the slide. When we look at a eukaryote, there are basically a couple of components to it. The first is the outer part of a eukaryote is a structure known as the plasma membrane. It's not a cell wall, it's a plasma membrane. And somewhere close to the center of the cell, doesn't have to be right in the middle in this picture, but it's not going to be way off in the corner, is going to be the nucleus. Now, everything between the plasma membrane and the nucleus is known as the cytoplasm. So that may be new to you. You may think that this yellow stuff here that I'm pointing to is the cytoplasm. That is not. That is the solution of the cell that makes up part of the cytoplasm, because the cytoplasm is everything between the nucleus and the plasma membrane. So this yellowish stuff is known as the cytosol. And in the cytosol, we will have floating various different organelles. So it's the cytosol and the organelles together that make up the cytoplasm. So when we look at the plasma membrane, this is what we are looking at. We're not looking at a little line being drawn on the outside of a cell. Here is when I say plasma membrane, this is what you need to be thinking of. And this is known as the fluid mosaic model. Fluid because, yes, it is fluid. It has the consistency of olive oil, which I know all of you know is a liquid. Okay, it's an oil. And mosaic, as in it's made of a lot of tiny little pieces stuck together. And the function of the plasma membrane is to control the passages of substances both into the cell and out of the cell. And by doing so, it creates a separate environment inside the cell. So the environment inside the cell is totally different than the environment outside the cell. And so we can use prefixes as in extracellular, as in meaning outside the cell, and intracellular, meaning inside the cell. So an internal environment of the cell would be an intracellular environment, and an external environment outside the cell would be an extracellular environment. Now, the plasma membrane is not a plastic Ziploc bag. It's not passive. It allows things to go through and it allows cells to communicate with each other. There is a lot happening in the plasma membrane. So we're going to look at it in a little bit more detail. First, it's made out of a phospholipid bilayer. So refresh your memory. We have a glycerol and two fatty acid chains to which we are going to attach a phosphate, and that gives us a hydrophilic head because it's polar and hydrophobic tails. And so when you put them in a watery environment, it spontaneously forms a phospholipid bilayer. So here is our cell with our phosphate-based hydrophilic heads, polar, lying external, as well as lying internal, and our lipid lying internal of the cell, but lying external of the bilayer, okay? And lying in the middle of the bilayer, or in the internal part of the bilayer, this is where the hydrophobic tails are, okay? And so this is where the lipid of the phospholipid is located. And on the two extremes is where the phosphate groups, the polar groups, are located. But it's more than just a simple bilayer. You can see all these swirls and other things attached to it. Right here, these structures, seen here in yellow, is cholesterol that is embedded. Notice the cholesterol is embedded in the lipid part of the phospholipid bilayer. And this cholesterol is a fat. So that is what's giving its rigidity, because cholesterol is solid at room temperature. Okay, it might be a pretty soft solid, but it's still a solid at room temperature, okay? This embedded cholesterol provides the rigidity in animal cells. But there is more that is embedded. There are proteins that are embedded, and these proteins have multiple functions. I want to show you that some of these proteins you can see right here. Okay, so the proteins in this particular picture are all shown as blue. And some of them, you see these little purple things attached to them. These are carbohydrate chains. And so when the carbohydrate chain is attached to a protein, then we can call it a glycoprotein. But when we look at these proteins in this phospholipid bilayer, we divide them into general types, these membrane proteins. The first type here are called either transmembrane proteins or integral proteins because they're completely embedded, or channel proteins because you see it has a channel in the middle. And these are the proteins that are going to regulate passage of materials either into the cell or out of the cell, because that passage, that channel, can close or open depending on messages and things binding to that protein. The second type of membrane protein can be either on the outside or on the inside. And this is showing one on the inside. And these are called peripheral proteins. So we had some here. Here's a peripheral protein on the inside. This is probably a peripheral protein on the outside. You can't really tell because we don't have a cross section at that area. And peripheral proteins can be attached to transmembrane proteins, or they can be attached to lipids. And some of these peripheral proteins are going to be enzymes. Some of these are going to form cell junctions to anchor cells to each other or to anchor cells to the underlying tissues. And some are going to be used for communications with other cells. So we'll learn more about this as the course goes on. So now that we have our phospholipid bilayer, don't forget that we have water both extracellularly and intracellular. And so small molecules such as water and oxygen can pass easily. Lipids can get through because we've got this lipid portion of the membrane. But some substances are going to need that channel to get through. And some are not only going to need the channel, but they may need a carrier to get through. And that's really beyond the scope of this course. That's something you would learn if you took a physiology course. Okay, so to end this section, what I would like you to do is to draw a plasma membrane and to label everything that you can remember. And I want to thank you for all your hard work in this class, and I will see you soon as a return and we look at what else is going on inside a cell.